So most of the work that I do now relates to studies of the HIV reservoir. So these would be the cells that are left over in patients who have HIV after you've successfully treated them with antiretroviral therapy. These cells are hard to get rid of. What my lab works on is trying to figure out ways, number one, to get rid of these cells using new strategies, but also to understand how HIV reservoirs in different parts of the body interact and what their relationship may be.
Our lab is now looking at the persistence of HIV in different reservoirs, ways to quantify the virus and to see how the amount of virus that we can measure relates to the risk of viral rebound in patients who interrupt therapy, and how different interventions may alter the amount of virus in the reservoir. We're also still doing some work on drug resistance, focused more recently on resistance to the integrase inhibitors and to some of the newer drugs that are coming along.
So I focus mainly on HIV persistence, looking at how and why HIV persists on antiretroviral therapy. For example, looking at stem cell therapies or cytoreductive chemotherapies for what the effect on the HIV reservoir, the amount of residual HIV after antiretroviral therapy, what impact that those have, and does this allow us to either stop ART intermittently, transiently, or even longer term.
I'm actively involved in the ACE clinical trials group, and at the same time I do run a translational HIV research lab as well. We're doing a lot of ACTG related studies there, looking at kind of viral and immune predictors of viral rebound, where we've identified a really remarkable group of HIV patients within the ACTG who are these post-treatment controllers who are able to control their virus even without, even after they stopped antiretroviral therapy, and we're trying to figure out how they're able to do that and whether we can apply some of the things we can find there and to help our other patients.